Okay, welcome to your Day 7 notes video for Unit 1, Chemistry of Life. Today we're going to be talking about proteins and nucleic acids, the last two biological macromolecules that we need to cover. We talked about carbohydrates and lipids last class, so we're going to start with proteins today and finish up with nucleic acids. And proteins are biological macromolecules that include a diversity of structures. There's many different types of proteins. So since proteins can take on a variety of structure, we see a wide range of functions associated with proteins as well. So some of the functions proteins can take on I have listed down here, so we're going to talk about a couple of them. The first thing that proteins can act as enzymes. So there are enzymatic proteins. So certain proteins will act as catalysts for chemical reactions. They help accelerate chemical reactions. We'll talk more about these in Unit 3 when we get into more detail and more, we do more of a deeper dive into enzymes. But just be aware, proteins can be enzymes. There are certain proteins that have defensive functions. Certain proteins protect against disease. Antibodies, for example, are one such case. There's also hormonal proteins. Hormonal proteins help coordinate organisms' activities. So something like insulin, that is a hormonal protein that helps regulate blood sugar levels. Proteins can also function as receptors, so they can help cells respond to chemical stimuli. Proteins can serve storage functions. Certain proteins can help store amino acids. Casein, the protein of milk, is a major source of amino acids for baby mammals. So there is a protein serving a storage function. Proteins can also help with cell transport, so transporting substances across cell membranes, for instance. There are contractile and motor proteins that help with movement. Actin and myosin are certain proteins in muscle cells that help our muscles do their thing, help move our bodies. And last but not least, proteins can also take on structural functions. So they can have a support role in the body. A great example of a structural protein is keratin. This is the protein that makes up hair. It's also found in horns, like rhino horns, for example, feathers, and that keratin is serving an important structural role, helping to maintain the structure of those hair, of that hair. Okay. So variety, again, of functions, and that has to do with the fact that we can have a wide variety of structures from our proteins as well. Okay, structure determines function, so a wide variety of structures translates to a wide range of functions. And then again, just a little note about enzymes, since those guys are some really important proteins we're going to talk about a lot throughout the year. Those are those proteins that act as catalysts to help accelerate chemical reactions. And what's interesting and cool about enzymes is they are able to perform their functions repeatedly. You can reuse an enzyme so long as that enzyme has not been marked for destruction. That enzyme can just keep on doing its job, functioning as sort of a workhorse in the cell, helping to carry out a lot of those really important processes of life. Okay. So keeping all of these diverse functions in the back of our mind, let's start talking about the structures of proteins. We do have a special name for the monomers and polymers of proteins, just like we do for carbohydrates. So the monomers of proteins are what we refer to as amino acids. Hopefully you remember that from first year bio. And proteins are all constructed from the same set of 20 amino acids. The way that we arrange those amino acids is what gives proteins their unique properties. We can have all sorts of different combinations from just 20 amino acids. Okay, so the monomers are amino acids. The polymers are called polypeptides. Polypeptides are our unbranched polymers built from those 20 amino acids. And if you are talking about a protein, when scientists mention proteins, they are usually referring to the biologically functional molecules consisting of one or more polypeptides. So a protein might consist of just one polypeptide. It might consist of multiple polypeptides together in a structure. So once we've folded up that polypeptide correctly, we have ourselves a protein. Okay. So for those polypeptides, in order to make a polypeptide, you are going to see a dehydration reaction. Okay, dehydration synthesis. There's that hydroxyl group leaving and that hydrogen there leaving from this amino group here, leaving this as a water molecule. And what's going to form in that place is a special covalent bond. So this is a covalent bond known as a peptide bond. So amino acids are linked together by these covalent bonds called peptide bonds. Each polypeptide has a unique linear sequence of amino acids. So right here, this is one amino acid, and then this is a different amino acid. They are different because of this little side group here. We'll talk more about that here in a second. But there's a unique sequence of amino acids, and each amino acid is going to have a carboxyl end, or a C terminus, and an amino end, or an N terminus. Okay? So C for carboxyl, think N, amino, okay? And the length of polypeptides will vary. There are certain polypeptides that are only a few amino acids long, and there are some polypeptides that are over 1,000 amino acids long. But we link them together via peptide bonds, and it is, again, a link between that C terminus and that N terminus. That is where the dehydration reaction will happen. Water will leave, a new peptide bond is formed, okay? So if you want to make a little note off to the side that this is dehydration synthesis, forming that covalent bond there, then please do so. So now we're going to flip over to the next page and talk a little bit more about these building blocks of proteins, these amino acids. So amino acids are organic molecules, and they get their names from the amino and the carboxyl groups that make up their structure, okay? So right here we have the amino group. We recognize this functional group, hopefully, from our previous discussions of functional groups as an amino group. And the carboxyl group over here is that carbon double bonded to an oxygen, and then it is also bonded to this OH over here, okay? Amino acids, they're all going to have the same basic structure. They have an amino group, a carboxyl group, and an H on the central carbon. The only thing that differs between amino acids is this side chain, okay? The identity of the amino acid is determined by this side chain. So amino acids differ in their properties due to differing side chains, which we call R groups, okay? You do not need to memorize the names or the structures of specific amino acids for the AP exam, okay? Like I said, there's a lot of them. These are all the different amino acids. You do not need to memorize those structures. If you go on to take biochemistry in college, they will probably make you memorize them. But we do not need to do that for this class. What you need to know is I need you to know how the properties of these different side chains are going to affect protein folding in general, okay? So what I mean by that is I need you to be able to recognize that these different R groups are different chemically, and since they have different structures, they can interact with water or not interact with water, okay? So when we start looking at side chains of amino acids, I need you to remember that water is a polar molecule. So if you're looking at one of these R groups, these side chains and these amino acids, charged molecules and other polar molecules will interact with water because water is polar, and nonpolar molecules will not interact with water. Nonpolar and polar do not want to interact, okay? So polar side chains and charged side chains are okay being in contact with water. Nonpolar side chains do not want to be in contact with water. that is going to affect how proteins fold. When we start talking about protein folding, when we get into primary, secondary, tertiary, and quaternary structure, the side chain properties are going to factor into how that protein ends up folding. Okay, so here's all of our amino acids. I want to point out, the different types. We have, of course, some non-polar chains. So you'll notice that these non-polar chains, I'm going to highlight them. You'll see a lot of carbons and hydrogens making up these non-polar side chains, okay? Those are the chains that don't want to interact with water. If you look at your polar side chains, okay, we've got a little bit different structure. You'll start to notice some oxygens and things. Some of them have sulfur. You can see like some NH2s, some oxygen in these side chains, okay? These are our polar side chains. These are hydrophilic. They're okay with interacting with water. Our nonpolar side chains are hydrophobic. They are not okay with interacting with water. And then we also have some amino acids that have a charge to them. So we have some positively charged amino acids. These are our basic amino acids. So this side chain here, you can see there is a positive charge associated with these side chains. Okay. Those are basic. These guys down here are negatively charged side chains. So these are okay with interacting with water. I'm going to put will interact with water. For both of these. And like I said, our polar are hydrophilic. These are water-loving, will interact with water. I'm just going to abbreviate with here. That's with water. And then our hydrophobic ones don't want to interact with water. So just like we talked about with the phospholipids last class, how the hydrophilic portions of the phospholipid are okay with interacting with water and will orient themselves towards water, hydrophilic amino acid side chains are going to orient themselves towards water. They're fine orienting themselves towards water, whereas hydrophobic amino acid side chains do not want to orient themselves with water. So when a protein goes to fold, that's going to have important effects on the actual ultimate structure of that protein, as these guys try to hide away from the water, and these guys are okay with being in contact with water, which is present throughout the cells. Okay? So the actual sequence of amino acids, the type of amino acids, and the order in which we link them together is ultimately going to determine how a protein looks, its 3D structure. A protein structure determines then how it works, because structure determines function, okay? The function of a protein usually depends on its ability to recognize and bind to some other molecule. So if it does not take on the proper structure, then it's not going to be able to take on the proper function, okay? There's a couple of different ways we represent proteins in diagrams. You may see some of these in your textbook. There's a couple structural models here. One of them is the space-filling model, so it looks pretty blobby. I'll zoom in a little bit. You can see this better on your notes page. It looks a little blobby with these little blobs representing atoms making up the amino acids that are, being strung together to make this protein. A ribbon model is another common model you'll see. One of the things that you'll start to see in 3D structures of protein are these like helical structures or sheet structures. So there's some like little sheet structures right here, kind of hard to see back here. And then there's like a helical structure right here. So ribbon models are really good at showing, where some of these helical and sheet type structures in the protein are. The other way you might see proteins represented in a 3D model is like with a wireframe. And this is just a little bit more detailed. It kind of takes aspects of both the space filling and the ribbon model to show the chemical structure, as well as some of the helices and the sheets that might be forming as we look at this 3D structure of this protein, okay? In your textbook, you also might see really basic drawings of proteins. They aren't necessarily all going to be super complicated 3D models. People who do information like research or look into the physical structures of proteins, they have really sophisticated computer programs that can help them model like this proteins in these ways. But you can also draw really basic diagrams to just represent what a protein is doing. Really simplified diagrams might just be solid shapes to show where a protein is, for example, in a cell membrane if we don't need a lot of structural details. They might even show a transparent version of this shape to show, what the internal details, some of the internal details look like. Like, for example, here, it looks like there's some helical structures within the internal structure of this protein embedded in this membrane that we see here, okay? A lot of times enzymes, we do real basic shapes to represent enzymatic proteins. So here, this simple shape is used to represent a generic enzyme, and these things are called substrates. We'll go over that when we talk more again about enzymes later. But we might show, where the substrates, the substance that the enzyme acts on, where that binds by showing these little indentations in this shape, okay? And then sometimes we can go super, super basic and just represent a protein with a dot. So this is an insulin-producing cell in the pancreas that is drawn right here, and we are showing that this insulin-producing cell releases the protein insulin, which is a protein hormone, from the cell, and they just represented that protein with a dot. So you can see the wide range of ways we can represent a protein from, very complex structural, space-filling, and ribbon models all the way down to just a little dot. So you'll see lots of different pictures of proteins as we talk about them throughout this unit and as we continue to talk about proteins the rest of this year. Okay. So, just a little note about protein structure and function. Let's get back into talking about actually how proteins fold. And again, we're going to keep in mind the properties of the R groups, whether or not they are hydrophobic, hydrophilic, or charged, to get an idea about how the protein may fold itself and ultimately end up looking like, the 3D structure it's meant to. So on the next page, we have our 4 levels of protein structure. We're going to talk about those here, and I have a lot of images to help visualize these because protein structure is a little hard to wrap our heads around sometimes. I have a actual 3D model I'm going to show y'all next class. So for my 8A students who are watching the video, I apologize. I'm helping out with a lab right now in another class. So I will show you that 3D model that I have in class during our review day, so you get to experience that. But for now, we're just going to do our best to look at these pictures and try and wrap our heads around what's happening in each level of protein structure. So for the first level of protein structure, we have primary structure. The primary structure of a protein is going to refer to its unique sequence of amino acids. The unique sequence of amino acids. So we can see here, this primary structure, it's what order are we putting the amino acids in as we link them all together to form a polypeptide. So this is like a little zoom in of these first 3 amino acids right here. It looks like we have glycine, proline, I believe that's threonine there. It's kind of hard to see. But there's one amino acid, and it's covalently bonded to the next amino acid over here, this proline, which is covalently bonded to this next one. So these peptide bonds are linking these amino acids together, and the order in which the amino acids are linked together is referred to as its primary structure. Okay. The secondary structure of a protein consists of what we call coils and folds in the polypeptide chain. The coils and folds, oftentimes called the alpha helix, is coil, and the folds, the beta pleated sheet, are going to result from hydrogen bonding between repeating constituents of the polypeptide backbone, okay? So this is hydrogen bonding between the polypeptide backbone. And in this image I included here on your notes, you can see the hydrogen bonding, represented with these dashed lines. Remember that hydrogen bonds are actually forces, and so we represent them with dashed lines. So I'm going to make a note there that dashed lines represent hydrogen bonds. And so here's our primary structure, is the actual sequence of amino acids as we're coming through here, right? That's the primary structure. That's the covalent bonds, the peptide bonds, and the order in which the amino acids are put into those peptide bonds. But you can see over here in this part, one part of this polypeptide is interacting with another part of this polypeptide chain via a hydrogen bond, okay? So when we start forming these like hydrogen bonds between these different parts of the polypeptide chain, the protein will start to form this alpha helix structure. And so the hydrogen bonding that forms between those backbones of the polypeptide is going to contribute to that secondary structure. Okay, same thing over here. We have these beta-pleated sheets. Okay, imagine I can extend this out. Maybe this looped back this way, and this is all still part of the same polypeptide chain. Okay, this hydrogen bonding, this is a hydrogen bond between one part of the polypeptide chain and another distant part of the polypeptide chain. They're interacting, forming a hydrogen bond, and that is what we call secondary structure. Okay, typical secondary structures, again, are those coils called alpha helices. So we can call that coil an alpha helix or alpha helices if we're making it plural. And the folded structures here that I just pointed out are what we call beta-pleated sheets. So this folded structure is a beta-pleated sheet. So alpha helix and beta-pleated sheets, these are part of the secondary structure. They arise from hydrogen bonding between the polypeptide backbone, okay? The repeating constituents of the polypeptide backbone. That is not to be confused with the hydrogen bonding that sometimes happens in tertiary structure, okay? So the next level of protein structure we're going to talk about is tertiary structure. Tertiary structure is going to be the overall shape of a polypeptide that results specifically from interactions between R groups. So there's where those side chains, those unique side chains of the amino acids come into play in protein folding. The R groups can interact with each other, and those interactions are of a wide variety, okay? Some of those interactions could be hydrogen bonds, okay? Just like there's hydrogen bonding over here, there can be hydrogen bonding in tertiary structure. The difference is this is hydrogen bonding between the R groups, okay? This is specifically referring to hydrogen bonding between the R groups of different amino acids. In secondary structure, the hydrogen bonding results from repeating constituents of the polypeptide backbone. So interactions between the R groups can include hydrogen bonds. They can include ionic bonds. Here's an example of an ionic bond. Okay, this positively charged group interacting with this negatively charged group. That's an ionic bond. There can be hydrophobic interactions. Hydrophobic interactions. So hydrophobic interactions and van der Waals interactions. Okay. These are interactions between nonpolar R groups. So this is a nonpolar R group interacting with another nonpolar R group. That would be considered a hydrophobic interaction at that tertiary structure level. You can also have certain strong covalent bonds form called disulfide bridges. Okay, so strong covalent bonds called disulfide bridges may reinforce the protein structures. These are called disulfide bridges. Di is that prefix that means two, and then the sulfide refers to the sulfur. This is the side chain specifically of a special amino acid called cysteine. It has sulfur in its side chain, and the sulfur can bond with another sulfur over in another cysteine R group and form what is called a disulfide bridge. So that's a covalent bond. So you can have all sorts of interactions happening at the tertiary structure level. Hydrogen bonding, ionic bonding, hydrophobic interactions, disulfide bridges. The important thing here is that they are interactions happening between R groups, okay? One R group to another R group. And it's kind of hard to imagine this using just this picture. I wanted to show this picture since it does include the hydrogen bond, the hydrophobic interactions, the ionic and the disulfide bridges here. But I wanted to show you down here in this image, as we go from primary to secondary to tertiary, what that polypeptide, what that protein is looking like. So remember that the primary structure is the sequence of amino acids. As the amino acids, as the polypeptide starts interacting with other parts of the polypeptide and forming hydrogen bonds between those constituents of its backbone, it can get into either alpha helix or beta pleated sheet in the secondary protein structure level. And then if you have like a long alpha helix or a long beta pleated sheet, okay, the protein will start to fold even further on into itself, okay? You can see how this is an alpha helix, but then the alpha helix itself is folded over in such a complex way like this. The reason why the alpha helix is folding over on itself like this is due to all these interactions with the R groups. So the sequence of amino acids and the R group sticking off from each amino acid will ultimately affect how the structure starts to fold over itself once we reach this tertiary protein level structure, okay? So that is tertiary protein structure, interactions with the R groups. The protein is folding itself even further. The last level of protein structure is not present. Not every protein has quaternary structure, but there are quite a lot of proteins that have quaternary structure. And quaternary structure, I forgot to underline tertiary up there, quaternary structure results when you have two or more polypeptide chains, forming one macromolecule. So if you get yourself a tertiary protein structure, and then you get another tertiary protein structure, and those two things start to interact and end up forming one macromolecule, we would say that that protein has quaternary protein structure. So there's an example of a protein with quaternary protein structure. You can see this piece, this tertiary protein structure piece here, and then you see another piece over here interacting with this to form one macromolecule. A great example of a protein with quaternary structure is hemoglobin. Hemoglobin is a globular protein that is actually made up of 4 polypeptides, so imagine like this, but 2 more of these tacked on. Hemoglobin is made up of 2 alpha subunits and 2 beta subunits, and hemoglobin is that molecule that is going to help carry oxygen in the blood. So a very important protein that has quaternary structure in our body. So you can just make a little note that this is an example of a protein that has quaternary structure. And then down here again with our structure of proteins, I want you to add a couple little notes to help you remember the difference, because visuals are just so important for this, since I feel like it's so hard to wrap our brains around how proteins are folding sometimes. I want you to remember that primary protein structure involves the sequence of the chain of amino acids. I want you to remember that secondary protein structure is going to involve alpha helices and beta pleated sheets, and this is due to hydrogen bonding between repeating constituents of the polypeptide backbone. And I want you to underline that tertiary protein structure. The way that the protein starts to fold in tertiary protein structures is due to side chain interactions, okay? AKA interactions between R groups. Okay, so those are specifically interactions between R groups. So some of these might be hydrogen bonds, but they're not hydrogen bonds between the repeating constituents of the backbone. They're hydrogen bonds between the R groups. And then, of course, our quaternary structure is the protein consisting of more than one amino acid chain, more than one polypeptide chain. Okay? So a protein with some side chains that are hydrophilic or charged may fold in such a way that those side chains are facing outwards of the protein, okay? Think about the proteins that are facing the environment of the cell. The environment in a cell is aqueous. The solvent in the cell is water, so there's lots of water inside the cell. The protein is going to fold itself in such a way that the hydrophilic or the charged R groups are oriented towards the water. Okay, so they might be on the outside of the protein, or if the protein is a funnel type protein, it has a barrel type function and is trying to let things through, there might also be some hydrophilic side chains hanging out in the middle. But when this protein folds, again, it's gonna fold in such a way that the R groups that are okay with interacting with water are in contact with water, and the R groups that are not okay interacting with water are going to kind of hide themselves away in the protein, away from the water. Okay? And again, I will show you a visual. I have a nice 3D model of a protein, and I can show you kind of what that's going to look like next class. That way you have a better idea of what's actually going on visually there. Hopefully this image helps you visualize these different levels of protein structure, though. So protein structure, the levels of protein structure are important because if you start changing something in one level of protein structure, oftentimes it's going to have a ripple effect and affect the protein in the higher levels of protein structure. So, for example, if we do something as basic as change the primary structure, we can change the ultimate quaternary structure of the protein, as that simple sequence change can cause a structural change profound enough to affect the folding of the ultimate protein that is being created. So sickle cell disease is a great example of a disease that results from a very basic one amino acid change in primary structure. In people who do not have sickle cell disease, in spot 6, they have this GLU amino acid, whereas those who have sickle cell disease, they have in spot 6 this VAL. They have a valine in that spot instead. And so what ends up happening is if you change this one amino acid in the primary structure, change the order, the amino acids making up the primary structure, for this case, for this disease, you end up changing the secondary and the tertiary structures as well as the quaternary structures. Okay? So this is an inherited blood disorder, and by simply just substituting this one amino acid, we have effectively changed the final structure of the red blood cell. Okay? The reason why sickle cell disease is called sickle cell disease is because the people who have this condition, they end up with these red blood cells that have this sort of sickle or half-moon shape. Okay? And this half-moon-shaped red blood cell is not as good at carrying oxygen as this red blood cell that we see in the normal phenotype. Okay? So the oxygen carrying capacity is reduced here. So this is a problem and causes people to experience symptoms associated with that inability to properly carry oxygen. So I want to make a little note here that the effects from the change in primary structure for this protein, the effects from the change in primary structure of this protein are seen all the way through quaternary structure here. And that's what happens with most disease states, is You're changing the structure in such a way that it is affecting the function of that molecule, that cell, what have you. Okay. So typically if you change, like if I were to change a secondary structure, for example, in another protein, that could subsequently affect tertiary and quaternary structure. A lot of times those effects are felt upwards as you go up to those higher levels of protein structure. There's also other things that determine protein structure other than just the primary structure. Okay, obviously the sequence of amino acids is important and is helping to determine how the protein will ultimately fold, or how the polypeptide will ultimately fold into a protein. But there's other factors in the environment that will affect the way a protein polypeptide chain folds as well. So in addition to primary structure, physical and chemical conditions can also affect protein structure. If you start altering things like the pH, the salt concentration, or the temperature of the environment, you can cause a protein to unravel. Most proteins, they like to exist within a specific range of pH, salt concentration, or temperature. And if you start pushing them outside those bounds, you start raising the temperature super high, for example, you can actually cause a protein to unravel. You can disrupt the chemical bonds and the forces within that protein such that it denatures. So the loss of a protein's native structure, when the protein unravels, we refer to that as denaturation. And a denatured protein is biologically inactive. It means it can't do its job. Denature a protein, it cannot do its job anymore because it's lost its structure. Proteins, in order to do their job, they have to have a specific structure, and so that polypeptide has to fold in a certain way to interact with other molecules in a certain way. And if it loses that shape, it can no longer interact with the molecules it's trying to interact with. Okay? So how do scientists know how proteins fold? How do we predict how they fold if we're trying to figure out, what is this polypeptide going to fold into, or what is this doing in the cell? Well, it is kind of hard to predict how a protein ultimately will fold from its primary structure. Most proteins probably go through several stages on their way to the stable final structure. There is a big field in biochemistry and in bioinformatics in general. People are very interested in determining how proteins fold and kind of how the sequence of amino acids determines how a protein folds. So researchers will do a lot of work looking at or trying to predict how proteins will fold. In addition to trying to predict how proteins will fold based on the amino acids and the properties associated with them, scientists can use things like X-ray crystallography to try and determine a protein structure. Nuclear magnetic resonance spectroscopy is another imaging method they can use. These are just some really interesting imaging techniques that help us visualize proteins. It doesn't necessarily require us to crystallize the proteins. Bioinformatics, like I mentioned, is a great way to predict how proteins fold. Many scientists and researchers are using computer programs to compile information about what they know regarding protein folding to help make predictions about how other proteins will fold. And protein folding is just super important in general to look at the actual functions of different proteins in our bodies, to look at different disease states. There's a lot of different diseases like Alzheimer's, Parkinson's, and BSE. This is short for bovine spongiform encephalopathy, which is mad cow disease. These are diseases specifically associated with misfolded proteins, and so it's of great interest to scientists to understand why proteins end up misfolding in certain diseases and why they, don't fold properly, or why they start to do things that we don't want them to do, because then we can then start to look into potential treatments for diseases. Okay, so that's our information on proteins. If you have questions, again, please ask me next class since you're just watching the video this class. But for now, we're going to go ahead and move into our last biomolecule, which is the nucleic acids. So nucleic acids, just like proteins and carbohydrates, they have their own special names for monomers and polymers. And so these guys are going to be really important for transmitting information from one generation to the next. And so we're going to talk about what these nucleic acids are made up of, and we're going to talk a little bit about the central dogma as well today. Just mention that, since you can't really talk about DNA or RNA, which are nucleic acids, without talking about central dogma. So the amino acid sequence of a polypeptide is programmed by a unit of inheritance called a gene. Okay, so we have some genes in our body. They're housed within the nuclei of our cell. So genes consist of DNA, which is a type of nucleic acid, which is a biomolecule that we're going to talk about today. Nucleic acids are polymers called polynucleotides. Okay. So the polymers of nucleic acids are called polynucleotides, and each polynucleotide is made of monomers that we call just nucleotides. So monomers are nucleotides, polymers are polynucleotides. Each nucleotide consists of 3 parts. And I always remember these 3 parts because when I was in AP Biology in high school, my AP Biology teacher had a little saying. I don't know why it stuck so well in my head, but it still sticks in my head to this day, so I'm going to share it with you today. And that's saying that each nucleotide consists of a sugar, a phosphate, and a nitrogen base. So again, it's a sugar, a phosphate, and a nitrogen base. Okay? So we're going to write that over here. Each nucleotide has a nitrogenous base, a pentose sugar, and 1 or more phosphate groups. A sugar, a phosphate, and a nitrogen base. If we're just looking at the portion of the nucleotide without the phosphate group, we call that a nucleoside, but we're usually just going to talk about them as nucleotides. Okay? And so here, for example, is a nucleotide with its sugar, its phosphate, and its nitrogen base. Now, there are 2 different families of nitrogen bases, and I want you to be familiar with them, the purines and the pyrimidines here. They're pictured here. So on your next page, I want you to fill in for that first blank that one of those families is the pyrimidines. The pyrimidines are cytosine, thymine, and uracil. These guys are made up of a single 6-membered ring. So if you look over here at the chemical structure of cytosine, thymine, and uracil, they have one 6-membered ring. So they look like little hexagons here. Okay? Those are our pyrimidines. The purines, are adenine and guanine. They have 6-membered rings fused to a 5-membered ring. So purines are made up of 2 rings. Oops. I'm going to pause this video real quick while the bell's going off. Okay. So back to the video. I was at purines. So I had mentioned purines are the adenine and the guanine. So they have a 6-membered ring fused to a 5-membered ring. And so there's our adenine and our guanine. So there have been some AP biology questions in the past that have have required students to know that cytosine, thymine, and uracil are these single-membered, 6-membered rings, whereas the adenine and the guanine have the 2-membered rings. So I have a way to help you remember that adenine, A, and G are going to be your 6-membered rings, or your purines are going to be your two rings fused together, and your pyrimidines are going to be your single six-member ring. They're kind of really weird memory devices, but they've worked for me in the past, and some students have told me they found them helpful, so I'll share them here. If you have a different memory device that works better, please tell me. I'm always on the lookout for new memory devices that can help y'all out. So my first memory device is purines. Okay, I remember that purines are A and G, okay, because A and G together, they're ag, okay? So ag is short for agriculture, and I always think of Purina, okay? Whenever I see purines, I always imagine the dog food brand Purina. I think they make other animal feeds as well, and so I think, okay, animals feeding animals, that's agricultural. So I always remember that my purines are A and G, again, because I associate this as like agriculture, and with Purina, which is, again, they make animal feeds. So that's how you can remember potentially that A and G are your purines, and then the other three are going to be your pyrimidines. The other way I remember, the other thing you need to remember is that your purines are going to have the two rings, okay, so the six-membered ring fused to the five-membered ring, and then your pyrimidines have the single six-membered ring. So the way I remember that purines just have the that pyrimidines just have the single ring and purines have the two rings is because pyrimidines is a longer word than purines, okay? So this is my memory device that doesn't make sense, kind of, but it makes sense in my brain. So you would think, wouldn't it be a great memory device if the pyrimidines, which is the longer word, were made up of the two rings, and the purines, which is the shorter word, is just the one ring. That's not the case, okay? But a lot of things in biology don't make sense, so sometimes my memory devices that don't make sense work the best because biology is just nonsensical sometimes. So if you remember that pyrimidines is the longer word, and so therefore we only have the one ring. It's kind of the opposite. The long word just has the one ringed basis here. And then the short word has the two rings. Our purines have the two rings. Our pyrimidines have the single ring. Again, it's kind of a really odd way to remember it, but that's just how I always remember it. If you have a better way to remember purines versus pyrimidines, again, please share with me because I would love to hear your device as well. So there's my little tangent on purines and pyrimidines and how to potentially remember them. Again, pyrimidines as single rings, and purines, they've got the two rings. So if you want to draw your little two rings there and your one ring there to help you remember the difference, then please do so. Okay? In this next blank, I want you to write the difference in sugars between DNA and RNA. So the two nucleic acids that we typically talk about are going to be your DNA and your RNA. And in DNA, the sugar is going to be deoxyribose. So that ose ending, again, shows up with the sugar. And in RNA, it's just going to be ribose. Okay? So we've got our nitrogen bases, we've got our sugar, and we have a phosphate group as well. Okay? You do not need to be able to draw out the specific structure of specific nucleotides. They are never going to ask you to identify like this as cytosine or thymine, for example. They would never give you these two and be like, which one's cytosine, which one's thymine, okay? No, I do want you to generally know the difference between pyrimidines and purines, but I don't need you to draw these structures out. Okay? And then I went ahead and included this picture here. This is a really famous picture that was taken by Rosalind Franklin. You can see Franklin, her name right there. This is what is called Photo 51. We got a whole bunch of information about DNA. This is what ultimately helped us realize DNA's structure back when scientists were first trying to puzzle that out. That picture was key in giving us a lot of really important information about the spacing between base pairs, the minor groove and the major groove, and some other things that we'll talk about later in this course as well. So those are, again, nucleotides. To kind of recap what we talked about here, our nucleotides are going to be our monomers, and each nucleotide is made up of a sugar, a phosphate, and a nitrogen base, okay? Our polynucleotides are going to be when all the nucleotides are linked together to form our polymers. And how we link nucleotides together, we do have a special name for the bond, just like we do for carbohydrates and proteins. Carbohydrates, we linked monomers together with a glycosidic linkage. Proteins, we linked them together with a peptide bond. Nucleotides are going to be linked together by something called a phosphodiester linkage or bond. The phosphodiester linkage or bond is what links those nucleotides together. It's a covalent bond that holds together polynucleotides. And these linkages consist of a phosphate group. So there's one of those functional groups we talked about. They consist of a phosphate group that links the sugars of two nucleotides and creates a backbone of sugar phosphate units with nitrogen bases as appendages, okay? So if you look on the previous page, page 29, you can see this sugar phosphate backbone right here. I'll highlight this in yellow here. It says on blue background here, but you did not have notes printed in color. So I'm just going to highlight it in yellow. That is your sugar phosphate backbone, okay? A little hard to see. But that little pentagon-looking thing, that is the sugar. And then you've got some little phosphates represented by these little open circles here, okay? So we've got our sugar phosphate backbone. It's alternating phosphate, sugar, phosphate, sugar, phosphate, sugar, okay? So these are your phosphodiester bonds. Focus that a little bit better so you can see that, sugar phosphate backbone. Now we'll zoom back out. Continue talking about that, okay? So the sugar phosphate backbone is going to be this part right here. For example, there's our sugar phosphate backbone. There's another sugar phosphate backbone. DNA is a double-stranded molecule, so it's got both those strands there. The sequence of bases along the DNA or RNA or mRNA polymer is going to be unique for each gene. And so you'll see there's this backbone, and then we have these nitrogen bases sticking off as appendages. The sequence of those nitrogen bases will be specific to that specific gene. Okay? And so when we start talking about the structure of DNA molecules versus the structure of RNA molecules, we're going to refer to this picture to help us see some of these visual differences. DNA molecules, these guys have two polynucleotides spiraling around an imaginary axis. So this is the classic double helix shape associated with DNA. So DNA's double helix comes from those two polynucleotides spiraling around this imaginary axis. The backbones that make up these polynucleotides, they actually run in opposite, what we call 5' to 3' directions from each other. So 5' to 3' is referring to the carbon here. So we call this the 3' end because this is considered carbon number 3 in this sugar right here. And then we consider this to be carbon number 5 up here. And so this would be the 5' end because the 5' is free up here and the 3' is free down here. So those are your 5' and 3' ends. So DNA molecules, they have backbones running in opposite 5' to 3. So the free 5' end up here on the other side of the DNA molecule, that other strand, you would find a free 3' end. So since they run in those opposite 5' to 3' directions from each other, we say that the DNA strands run anti-parallel to one another. And the two backbones themselves like the way that these two backbones are held together is because the nitrogen bases are interacting with each other via hydrogen bonds. So our two backbones here are held together by hydrogen bonds between the nitrogenous bases. So over here, again, we see some more dashed lines. These dashed lines represent hydrogen bonds, just like they've continued to represent hydrogen bonds throughout many of our images that we've encountered in our notes. Okay? So these strands are held together because, for instance, there's some hydrogen bonds between A and T. So A and T have hydrogen bonding. The number of hydrogen bonds actually differs depending on the bases that are bonded together. Only certain bases in DNA pair up, and then the certain bases form certain numbers of hydrogen bonds. So you may remember from first year bio that adenine, that big A, in DNA always pairs up with thymine. Okay? The complementary base of adenine is thymine. So A and T pair up. When A and T pair up, they form two hydrogen bonds. There's two hydrogen bonds linking the adenine and the thymine. The other complementary base pairs in DNA are G and C. So that's guanine and cytosine. So guanine pairs with cytosine, C, and when guanine pairs with cytosine, 3 H bonds are formed. Okay, so there's 3 hydrogen bonds between guanine and cytosine. You can't see in this picture, we don't show the 3 bonds and the 2 bonds, we just show in general that there's hydrogen bonding happening between these. But if you were to look at a more detailed picture of the structure of DNA, you could see the hydrogen bonding represented with 2 or 3 depending on the base pairs here. Okay. So complementary base pairing, the fact that A always pairs with T and C always pairs with G in DNA is called complementary base pairing. And that makes it possible for DNA molecules to divide and generate 2 identical copies whenever a cell is preparing to divide. So when your cell goes to divide and needs to make another copy of its DNA, this complementary base pairing is really helpful in that process. And we're going to talk all about DNA replication later this year when we get into like Unit 6, and you'll learn all about the enzymes and the process that is involved with that. But I did want you to be aware of the complementary base pairing, A's and T's, G's and C's. A, T, G, and C are found in DNA. There is a slightly different combination found in RNA. And so if you just know the nitrogen bases involved in the nucleic acid, you can actually determine whether it's DNA or RNA by looking at whether or not thymine is present. Okay? So let's talk a little bit more about RNA now. RNA, in contrast to DNA, is usually single-stranded. Okay? Not always, but a lot of times you'll find RNA in the body single-stranded, like single-stranded mRNA floating around. The difference between RNA and DNA, one of the major differences between RNA and DNA is that thymine is not present in RNA. Instead, uracil is the nitrogen base used here in RNA. So in RNA, A and U pair, okay? There's no thymine in RNA, okay? If you're ever asked, and College Board loves to ask questions about RNA and DNA and throw in the fact that, one of them has thymine and one of them has uracil. If you are ever asked, how many, if I have, 30% of my molecule is adenine in my RNA molecule, how much of my RNA molecule is thymine? Don't let that trick you because you know that adenine pairs with thymine in DNA. You need to know in RNA there is no thymine, there's only uracil. So there's no thymine in RNA, there's just uracil. There would be equal amounts, there could be equal amounts of A and U in RNA, but there might also not be, okay? Remember that since RNA is usually single-stranded, since it's usually single-stranded, A and U are not always present A and U are not always present in equal quantities. Okay? Same goes for G and C. Okay? Same goes for G and C. In RNA, since RNA is typically single-stranded, okay, there's no reason that the number of G's has to match up with the number of C's. Okay? In DNA, that is the case. Okay? In DNA, the number of G's is going to equal the number of C's, or it should equal roughly the number of G's, should equal roughly the number of C's. There could be some mutations or something happened, something went wrong. But most of the time, okay, G and C, the number of G's and the number of C's in DNA will be equal because they are complementary. There's two strands, and so they're always paired up together. They should have an equal number of G's and C's. In DNA, again, there should be an equal number of A and T because they're always paired up. Okay, they should always pair up together. There should be an equal number in DNA. But since they are not always paired up in RNA, since it's usually single-stranded, we're not always going to see that equal quantity. On the topic of pairing, I totally forgot to mention this up top here with DNA, but for those of you who don't have a memory device to help you remember that A and T pair up and that C and G pair up, I do have a memory device for you. I have a couple of different ones, actually. I always remembered that A and T pair up because A and T, when you write out capital A and capital T, you only use straight lines to write those out. So I always remember my bases that I use straight lines to write out go together, whereas my bases I draw curvy lines for, C and G, those pair up together. Another way you can remember A and T pair up and G and C pair up is you can use the phrase chips and guac. Okay, so chips and guacamole. So C and G go together because chips and guac go together. And T and A go together just because they're the other two that go together. I've also have, I know that a lot of people are really big fans of Taylor Swift. I have had students in the past tell me that they've used Taylor Swift lyrics to help them remember. So they've used, there's a Taylor Swift song I have been told called Getaway Car. So G and C, like Getaway Car, and then A and T, they, what was the other one? All Too Well. Yeah, so they had the Taylor Swift song All Too Well. They remembered A and T for that. So you can use whatever memory device helps you. If you just have it memorized, awesome. If you need a memory device, hopefully one of those three helps you. If you have another one, feel free to share with me. I'm always collecting memory devices to share with students, like I said. Okay, so just wanted to mention that real quick. And the last little thing we're going to do here is mention the role of nucleic acids in the body. So we've talked a lot about DNA and RNA. So what is DNA and what are DNA and RNA actually doing in the body? Well, nucleic acids, they're really important because they store, they transmit, and they help express hereditary information. So this is how hereditary information, genes get passed on to the next generation. That information is stored in those nucleic acids. And that's part of what makes you, you. That and gene expression, which we will learn more about in Unit 6. Okay. So the two types of nucleic acids we just talked about were DNA and RNA. So on the next page, if you want to write out the full name for DNA, DNA does stand for, oops, I did not show you that. There you go. DNA does stand for deoxyribonucleic acid. And RNA stands for ribonucleic acid. Okay. Remember that DNA, because it is able to complementarily base pair, it provides directions for its own replication. DNA is super neat in that it is a self-replicating molecule. Some enzymes will help DNA replicate, but it contains the instructions for its own replication by nature of its structure. And then DNA directs the synthesis of messenger RNA or mRNA, and through mRNA, ultimately controls protein synthesis. Okay, this process is known as gene expression. We have a whole unit, Unit 6, that deals with gene expression, so we'll get into the nitty-gritty of that later this year. But for now, I just want to mention the general flow of genetic information since it's super important for biology. And that is, from DNA, we can make RNA, and we can use RNA as the instructions to build proteins, which are the other biological macromolecule that we talked about today. And proteins do all sorts of things in our body, as we saw from the variety of functions mentioned at the beginning of our notes. So hopefully we're feeling like we know a lot more about proteins and nucleic acids now, their different, what their monomers are, what their polymers are. What I went ahead and did for y'all is I made y'all a chart down here for each of the 4 biological macromolecules: carbohydrates, lipids, proteins, and nucleic acid. And I wanted you to write the elements that we can find in each. For example, I'll start you off with carbs. Carbohydrates contain the elements carbon, hydrogen, and oxygen, okay? And roughly a 1-to-1 ratio. The monomers are always a 1-to-1 ratio. The polymers are roughly a 1-to-1 ratio, okay? You don't need to put the ratios for any of the other ones. I just thought I would mention that for carbs. But tell me what the monomers are for each of these biomolecules. Tell me the type of covalent bond linking the monomers. There's a special name for each of these. I didn't talk about this one, so if you want to write that fatty acids, you don't really need to know this one, but fatty acids do attach to glycerol via an ester bond, just so you can have that in there, okay? But you know the other ones. We talked about the other types, the specific name for the covalent bond linking the monomers. We talked about some of the functions that each of these biological macromolecules are tied to, and we also talked about some examples of each, okay? We've got at least a couple examples of each that were mentioned. So do your best to fill this out. I'm going to post this key for this summary chart on Canvas so you can check yourself when you're done, but I thought this would be a really good way for y'all to organize this information and hopefully start to review, because you need to be able to differentiate between them and know some of the major properties in order to be successful on your upcoming assessment. Okay, let me know if y'all have questions, and I will see you next class.